I'm Bruce Bugbee, professor of crop physiology at Utah State University and also president of Apogee Instruments. I'm here today at the Apogee Instruments studio to talk about our recent research on far-red photon effects on plant growth and development. This is an emerging field that's red hot right now. Companies are putting far-red LEDs into fixtures, and it's based on our work and that of some other laboratories that have clearly shown some value of far-red. So in this seminar, I'm calling this far-red, the forgotten photons. We can't see them, they're barely visible to the human eye, and yet they have powerful effects on plant growth that we're just discovering. We missed these effects for so many years because electric lights don't have far-red. There's no reason to put them in there. Sunlight is loaded with far-red photons, but it wasn't until we got LEDs, and especially a far-red LED that's very efficient that can be added to electric lights. So this work is both fascinating from the standpoint of fundamental photobiology and also has immediate significant commercial implications because we can manipulate both photosynthesis and plant shape with these far-red photons. So I'd love to show this prism breaking up the colors of light. Those of you know Pink Floyd, you know where this came from. So the far-red, I like to show this, it's right on this edge, right here, of just the edge of visible light. And that's what we're going to talk about in this. I want to acknowledge two of my colleagues that have done this research. They're in the trenches day to day making the measurements, and we meet every single day, discuss the data, what the next studies we're going to do. The first one is Shuyang Zhen, who was started as a postdoc. She's now a research scientist in the lab. She has done the measurements on photosynthesis, and especially canopy photosynthesis. And then also Paul Kusuma, who came to us from the University of Florida, Kevin Folta's lab there. He's done the research on plant shape from far-red photons. Our work has been funded primarily by NASA. They've funded the lab for over 30 years for some very basic studies, and now more recently, the USDA has funded our research through what's called the LAMP project, Lighting Approaches to Maximize Profits. Basic funding agency, applied funding agency, and the interaction between those has really helped us understand the value of far-red photons. Let me show you another graph of where the far-red photons lie. Here's the colors of light all the way from blue to far-red, and you can see, let's put some dotted lines on here, the cutoffs. Now, our traditional definition of photosynthesis doesn't start counting photons until, let's get some black, until right here. Right at 400, we start counting them. We have evidence these photons are useful too, but for now we're going to talk about just the far red. So right here on the edge of our vision, we're talking about these photons right in this area, right here. What's the effect on plant growth? Just to review, we typically break up light into three categories: blue, green, and red. And I'll be talking about the effects of those. It turns out these are 100 nanometer increments, but it really works well to break them up into this. We're currently studying different wavelengths of blue, but the green, they could look at the green includes yellow transitioning to orange. We call that whole thing green, but it turns out it works really well to break the light up into this. So, what I'm going to start with is a story, and it's a story of a study that Shuyang did with 4 growth chambers where we did canopy photosynthesis on the plants and gave them different colors of light. So let's go to that story. Here's the 4 chambers. 2 of them had white light from white LEDs. 2 of them had magenta or purple light from red and blue LEDs. Here is the input of photons to each. 300, 350, 300, 350. So if we looked at this chart without prior knowledge of far-red photons, how would you say the lettuce grew? This is a study with lettuce. The first thing you'd say, let's see if we can show this in red, is that these, by virtue of having higher light, would have higher yield. And these would be less. And if you were a believer in red and blue photons for photosynthesis, you would say that this would grow the best, and this would be second, and these would be third and fourth. Now let's look how the data comes out for this. These 2 were exactly equal. Now I've normalized the fresh weight and the dry weight to 100 for these to make the point. There was no difference in growth when we went from purple or magenta light to white light. That should not be a surprise, because now we know that green photons are just as good as any other color for photosynthesis. And I'm going to discuss that more in a bit. Let's take a look just at those 2 chambers and the difference. These 2 chambers grew even better in this experiment. So suddenly, look at the difference, 27% better, 35% better. This is just opposite of our intuition, where we thought this would be the best and this would be the worst. It's completely flipped. Let's take a look at what's going on here. Is there a mechanism for why this should be the best and grew better than that? What I didn't tell you is that these 2 chambers had 50 micromoles per meter squared per second of far-red photons. That's a big deal. So 300 plus 50, this got 350. The total here is 350. All 4 chambers had the exact same photon flux if you count the far-red photons. So what's up with that? Do far-red photons cause photosynthesis? Let's take a look at this. First, let's take a look at these 2 chambers. Is this right? Do white lights grow plants equally well to magenta lights? We have a long history of people saying pink houses with this blue. People say this is a plant growth light. This is not efficient for photosynthesis because the green photons just bounce off. Let's take a look at that. This is a 2002 edition of the plant physiology book that I use in class. What does this book say about red and blue and green light? 2002, that's 17 years ago. Here's the graph. Here's what it says. This is absorption of chlorophyll. Really strong absorption in blue, less in the green, strong absorption in the red. It says chlorophyll absorbs minimum green light. That's a true statement, and that resulted in people making lights with just red and blue because of this powerful absorption by chlorophyll. But sunlight is loaded with green light. Is that really wasted? Let's jump ahead to the 2015 version of this book, which is the current version. This is Thais and Zeiger. Now it's on the 6th edition. Now we turn to the exact same page of chlorophyll. Look at this graph. There's new pigments in here. And then now it says multiple pigments absorb radiation from 400 to 700. Now, this is still a little misleading because this pigment phycoerythrin is only found in algae. But the chlorophyll absorption, even though it's low in here on this graph, is enough to penetrate the leaves and cause excellent photosynthesis. So what I'd like to show you briefly is the effects of these colors of light. This summarizes a big part of my career to categorize these. Remember, we got blue, green, red, and now far red. Summary of spectral effects. Here's our dark side of the moon spectra. Blue photons inhibit cell expansion. That sounds terrible, but when it's stem elongation, it's a good thing. More blue, shorter plants, and that's tremendous. Now, if it's smaller leaves, that's bad. But that's the most fundamental effect of blue photons, is to inhibit stem elongation, but they also inhibit leaf expansion. Green are far more valuable than we once thought because they are very effective for photosynthesis and they facilitate human vision. Let's go to this slide right here. This is cannabis grown under red and blue LEDs and white LEDs. Our experience working in the lab is that these are tremendously valuable for diagnosing the plants. Get in there to see microscopic insects, subtle nutritional disorders. They penetrate deep in the canopy. We can see these dark areas. This is very difficult to understand the health of the plants. Now you can turn those off and turn on whites, but you don't see surgeons working under dim white light. They have bright white light, and we have found that really helps diagnose subtle disorders on the plants. This is still slightly more efficient electrically, but the difference is getting smaller. This is maybe 10% more electrically efficient than this, but this has the clear advantage for being able to diagnose the plants. This is a little sidebar from our quest to understand far red photons, but it's an important sidebar. Back to this. Inhibits cell expansion. Look at far red photons. Wow. They do the opposite of blue. They enhance cell expansion, and that effect is so powerful. Here's LED lettuce with white light, with no far red, and with 10% far red. Now, I just jumped ahead of myself to show you what's going on in our study with those far red photons, but this enhancement of cell expansion is a very big thing. It's a very powerful tool. Look at how well photons penetrate leaves. This is a beautiful study by Brodersen and Vogelmann, 2010. They looked at a side view of a leaf. Here's blue. There's so much absorption by chlorophyll, it doesn't penetrate. Here's green, penetrates the whole leaf. There's the top of the leaf. Let me get a green color here. There's the top of the leaf right there. Here's the bottom of the leaf. Green beautifully penetrates the leaf through the spongy mesophyll all the way through the leaf. That's a big value of green light. Red, similar to blue, absorbed by chlorophyll, absorbed in the top layers. Look at far red. That also penetrates the leaf. So these effects only become apparent when we study whole plants and especially plant communities. But this is a nice slide showing the penetrating effects of these colors of light. Back to our study. Better growth here, better growth here. What caused that better growth? When we get a result like this, that's sort of unbelievable, because if these don't cause photosynthesis, what's going on? We need to know the mechanism. And this, by the way, this is fresh mass and dry mass both in this study. We very carefully measure this. We've done it more than once. We get the same result. Now let's go back to Carl Sagan, who popularized a phrase that I think all of us would be good to remember when we're doing these studies. Extraordinary claims require extraordinary proof. With far red enhances lettuce growth, we need extraordinary proof to begin to understand it. So this comes from, first of all, the McCree curve. Here's a picture of Keith McCree. He died 5 years ago. Here's his famous curve. Here's 700. And in the McCree curve, he shows these cutting off so fast that these photons are of minimal value. But as terrific as a scientist as McCree was, and I got to know him personally early in my career. He was a physicist that came across to help biology. He was a great guy. He only had primitive tools to make this curve. He didn't have the LEDs we have now. He had prisms and filters. So we had low light levels, and he only could study one color at a time and on a single leaf. It's a terrific piece of work, and his papers are a masterpiece of writing, but they're limited because he couldn't study synergistic effects of multiple colors of light at once on the leaf. So let's look at what happens. There's our far red. This is a diagram of what happens with photons flowing through the light reactions of photosynthesis. Now, usually this is shown with a lot of biochemistry, but I really like physical diagrams. So here's a person, it's a man-wife team, I guess, moving photons through what we call photosystem 1 and photosystem 2. They first come in, it takes 4 photons to excite these 4 electrons to a higher energy state. And this is where water is split in photosynthesis. Now they get this photon, roll it down this platform, and this mill turns, and it makes ATP. So that's his first step, really valuable. Now they get to another platform, and we need 4 more photons to excite 4 electrons up here to this guy. There's 4 electrons in a very high state, and now we put them in a bucket, and this makes NADPH. So the combination of ATP and NADPH, very high energy compounds, are what causes photosynthesis. Interestingly enough, because of the way we discovered these, photosystem 2 comes first, and then photosystem 1. That's a trick question for students on plant physiology exams. 2 comes first. However, we have known since the 1950s and the time of Emerson that far red photons also are effective right here. And in fact, that work helped us understand there's 2 photosystems. So the far red photons can substitute for the traditionally photosynthetic photons here, but only if we have, we'll call it white light photons on this side. Far red photons all by themselves are not effective. We know that, but it's easy to give far red photons synergistically with all the rest of the photons. So this is a nice, easy to remember diagram of how photons turn into energy, and then all this energy is used to fix CO2 into sucrose in plants. But the point is far red photons. Now, there's a very fundamental law that underlies all of this, and this law relates not only to photosynthesis, but it relates to solar panels, and for that matter, it relates to human vision in the retina of our eyes. And that law, this is the Emerson enhancement effect. This law is called the Stark-Einstein Law. Stark helped explain Einstein's work. They both get credit for it, and here it is. 1 photon excites 1 electron. A simple law. We have 4 photons here, we get 4 electrons here. 4 more photons here, we get 4 more electrons up there. That's a remarkable law, and it links the photosynthetic value of photons to human vision and to solar cells and really all of biology. This is how the system works, and this is why far red photons can act synergistically with white. Now let's take a closer look at this. Is our definition of photosynthetic photons correct? To do this work, we put lettuce in those gas exchange chambers that I talked about and gave them supplemental far red light. This is just exactly how we did the study we're talking about. Here's the traditional photons. Here's the far red, right there. We normally wouldn't count them. For photosynthesis, what happened? This is 400 to 700. There's the far red. When we gave them only white light, we get this kind of curve for photosynthesis. This is gross photosynthesis, a curve that slightly nonlinear because we're getting up to high light levels. Now we start adding far red photons to this. What happens? Whoa, look at this. They go up. We're not counting them, but they made photosynthesis go up, and we're not counting them as photosynthetic. How do you explain that? We need another graph. Here is the graph where we count photons all the way to 750, and now those photons fall perfectly on the line. This is powerful evidence that our definition of photosynthetic photons needs revision to count far red photons, particularly in canopy photosynthesis, where they penetrate the canopy and they have a bigger effect than they might have in a single leaf. Let's take a look now at, does this happen in all species? Remember, we want convincing evidence. We want extraordinary proof. Maybe that only works in lettuce. Shuyang Zhen has now done this in something like 20 species: spinach, basil, kale, different kinds of lettuce, C4 crops like corn, sunflower, wheat. It happens the same in every species. Every single species, those far red photons are equally valuable to traditionally photosynthetic photons. Here's the commercial implications. We did a paper end of last year. It got published in the journal Nature. Most people show the flux of photons like this in an energy basis, and here's far red right here. Not very good. But energy doesn't cause photosynthesis. Energy doesn't cause plant shape. It's photons. And these are low energy photons. So when we take this data and regraph it for photon flux, look at this. The far red photons are almost equal to the reds and the blues. These violets are coming on fast too. That's a subject of another talk. But the far red photons are very effective. So now we can use those commercially. Not only can we use them for photobiology, but we can use them commercially to manipulate plants. Back to the study. Now this shows, if they're photosynthetic, this shows why these should all be equal, because they cause photosynthesis, but they weren't equal. These two are even better than those two. This gets us in to plant shape. This is Paul Kasuma's work now, and this is the response of plants to shade. When the plants are in the bright sun, they have plenty of visible photons, blue, green, and red. They know they're in the sun, but when they're in the shade, over here underneath this tree, they have a lot of far red photons. This is the graph of the shade. They're very enriched. When plants perceive a high amount of far red, they panic. We're under shade. They need to do one of two things: get tall fast to get into the light, or get bigger to capture more light. So there's two types of plants. Here they come. One's growing fast. This is called a shade avoider plant because it's getting tall. This plant is called a shade tolerator plant because it's getting big. If we can trick plants that are not in the shade to think that, we could make them like this. This is a traditional plant in the sun. This is exactly what happens with lettuce, that the lettuce gets big like this because of it being tricked by far red photons. Let's take a look at this. Shade avoidance, shade tolerance. There's a big picture of that lettuce and the value of far red. This was with just a little bit more far red. We tricked this lettuce plant into thinking it was in the shade. It got enormous. This is a powerful picture. We're all anxious to use far red for indoor agriculture, on lettuce to get the plants bigger. And many people have said this is incredible. We need to be giving far red to everything. But remember that far red also increases stem elongation, and some plants could be shade avoiders. Let's look at what the results from multiple species. Here's a top view of these plants. No far red, far red. It's the same slide as the last one, only shows you the dramatic benefit of far red photons, both because of photosynthesis and because of expansion. But look, the photosynthetic photons were less, so we needed those for photosynthesis, but they not only grew equally, they grew better. Here's the problem. We give the same amount of far red photons to tomatoes. Look what happens. More stem elongation. This is very undesirable in tomatoes. Well, I know we don't grow soybeans in controlled environments, but they're a great test species. It made soybeans panic. They get tall. This is not a good effect. Cucumbers makes cucumbers tall. This is not a good effect. Spinach, we have some evidence as leafy greens. This can help with leaf expansion in spinach too. So for leafy greens, the addition of far red is great. But it's a powerful tool because we could use it a little bit when the plants are young. We can use it at different times. Look at this flower right there. Let me see if I can highlight this. We'll put that one in red. There's evidence that far red light can help plants flower sooner. So I hope this seminar has helped explain from our results the tremendous value of far red photons. So historically, we have quantum sensors to measure photosynthetic photons, but those quantum sensors don't measure this far red. In order to measure it, you need a spectral radiometer, which is terrific, but that jumps up the price and complexity considerably in making those measurements. Because of this, Apogee has developed a sensor that has both red and far red detectors in the same sensor. So this helps us measure traditionally photosynthetic photons and in a separate measurement, far red photons. That's it. Thanks for listening. We'll talk to you again in another video.